So I'm Stephen Smith. I'm going to talk about some work that Joseph Brown and I have been doing in relation to the Open Tree of Life project. And I already feel bad because I said comprehensive seed plant phylogeny, but there's no fossils, so I'm sort of starting this off feeling pretty bad. But nevertheless, in my lab, generally over the last few years, we've been doing a lot more in phylogenomics and in genomics, so I kind of got away from large phylogenies, but for the Open Tree of Life project. And so, so here I'm trying to suggest some of the compromises that we've taken to try and add branch lengths and get a bigger, better view of land plant phylogeny. So bear with me, and yeah, let's see what we can do. So primarily we want phylogenies that are accurate and precise, up to date, and if possible have branch lengths relative to time, and comprehensive if possible. So the Open Tree of Life was aiming to do at least a lot of this work, and it's already been talked about a few times, but it's a fairly large project in terms of the number of PIs, but also the goals. And the idea was to synthesize existing data sources, not generate new data, to construct a tree of life. And there was a publication in 2015. There have been some updated methods by Mark Holder and Ben Redlings, and this project's still going. But the publication had a 2.3 million tip tree, and this is one view of it. These computers seem to be rendering in 1997, so it might not look as good as it could possibly look. But nevertheless, this is one view of this tree. This does not have branch lengths, and therefore it has some limited use for the community, but you can still do some things with it. So for example, I'm at University of Michigan, so I've been taking the plant, or the species list that we have for counties, and then other coordinates, and then making phylogenies of them. So for example, University of Michigan is in Ann Arbor in Washtenaw County, so this is a phylogeny of the Washtenaw County plants based on the Open Tree of Life. You can do that across the whole state. You can make more meaningful grids instead of counties, which are more political. Nevertheless, I want to show how we could try and add branch lengths and divergence times to this so that we could possibly make it a little bit more useful for other researchers. And something that would be sustainable so that we could do this continually. So as I've mentioned, the Open Tree of Life has some benefits. It's comprehensive, or nearly so. It's limited by taxonomy and some other resources, but nevertheless. It can be updated. And in some of the well-sampled areas, it has reasonable and good resolution. Some of the limitations are that the resolution may be poor, either non-existent or wrong, because the resources we have aren't necessarily the most up-to-date or accurate, or we don't know, because the community hasn't put their voice in yet. And also, it doesn't have divergence times or branch lengths. So, in the lab, we aimed to try and address this in the best way that we could think of. And again, with the idea towards sustainability. So something that we weren't going to do, millions of CPU hours and then that's it. Something that we could try and keep up to date as data continues to get added. So we developed a little bit of a technique that I'm going to go over, and this is implemented in Python and something called PyFlawed, based on some previous work that I have called Flawed. I didn't come up with another self-deprecating name, so PyFlawed is what you get. Nevertheless, the idea is to combine GenBank with OpenTree to get some resolution. Okay, so roughly the procedure goes something like this. We get a list of clades. So in the case of plants, this is pretty easy because the taxonomy and the phylogeny have been pretty well aligned for a while. So I happen to work on plants, but it also happens to be a pretty good test for this. Then for each of the clades, let's say roughly corresponding to linear orders, we can cluster and identify homologs from GenBank, construct phylogenies, and in some cases, assuming some monophyly that I'm going to go over, we can test these monophyletic constraints, but anyway, again, I'll go over that in a little bit. Try and estimate some support, the overlap of the data that we have, and then do some divergence time analysis. Then we can take these individual clades and add them back into some backbone. So that backbone can be open tree, which is our goal here, or some other tree. So for example, there's a tree that's come out of Magillon's lab that does divergence or has nice divergence times across angiosperms, so we can do that. We can also try and construct our own. So I said we do some genomics and phylogenomics in the lab. So work with my grad student Joe Walker is to kind of combine the construction of the deep edges and the tree and the tips at the same time. Okay, so we basically procedurally create folders that correspond to some sort of constraint tree. Okay, so in this case, just for illustrative purposes, let's say it's taxonomy. So you might have again, we're in seed plants, we have pretty good taxonomy. So maybe if you're talking about Linnaean groups, which we don't necessarily have to, but let's just say that maybe this folder is some order, family, genera, something like this. Okay, so each of them represents some putative monophyletic group, but they don't have to be monophyletic at the end of the day. They're just some putative group. There's some way to organize the data. Then we populate the folders that would be the tips of this with the data that's in GenBank. And then we do some kind of standard clustering techniques. So again, we do some phylogenomics, so we're kind of using those techniques to basically cluster this data. And then we kind of move back in this tree in a post-order fashion, and then cluster the clusters together as we move back in this case. So we might be left with orphan clusters that don't cluster with anything, that are stuck here, and those just get pulled down. We might combine clusters into these folders, and those go and populate at the base folder there. Now, optionally, we can break up clusters later on, so we generally join clusters. If we build phylogenies, find that they're poor phylogenies, and break up these clusters. I can get into that detail if anybody's interested. Okay, so then to construct the data set, we go and look back at this and look at each cluster and say, does this have reasonable taxon sampling? And then add those clusters to some supermatrix in order to construct a phylogeny. So for example, here is a supermatrix. And in some cases, we have some genes that are really well sampled, broadly sampled across the group, and some genes that are in one single plate. So we can accommodate both of these. Okay. So also just for your information, there's a folder for each of, there's a website that gets created as this, and you can click through and see where the clusters are in each of the folders. Kind of like Phylogenetic. Okay, so some edges are impossible to reconstruct without genomic data, or otherwise they're difficult to disassemble. Otherwise difficult to assemble data sets like genomic data sets. So one question is, can we assume monophyly? So do we have to confirm in each run that seed plants are monophyletic? That's a question. So we generally err on the side of no, we don't have to do that. We're going to assume they're monophyletic, construct a phylogeny, and then test those monophyletic constraints, and remove the constraints if they fail a test. In this case, we're using this Quartet concordance measure. It's similar to ICA, if you're familiar with that from Salakos and Rokas. And then here's an example of a constraint tree, and then one that has the constraint removed after a phylogenetic analysis. So here's a specific example. Valerianina is a clade that has not been recircumscribed. It is non-monophyletic. You assume monophyly. This is the MRCA for the forced monophyly. And then we test and break it up, and here's the MRCA, and it's paraphyletic. So this is just an example of a case where we know it's a non-monophyletic, and then we break it up. Okay, then we construct these trees and place them back into phylogeny. So on the left, we have the open tree. On the right, this is one clade, and then replacing it back in. So what do we get? So if we don't add taxa back that are not in GenBank, this is a tree that has 759, too many 7s and 9s, samples from GenBank with 61 unresolved nodes. So that's pretty good. There's a lot more to come. So we can also add unsampled taxa back that are in open tree that are not in GenBank. So if we do that, we can leave the resolution also that's in open tree. We get a tree that has over 356,000. That's pretty good. That's pretty close to seed plant diversity we would expect. So now you see we have 272 unresolved nodes, so a lot more work to do. Now we have to do divergence time estimation. So we use Magillon et al. 2015, which has a lot of dates for basically Linnaean families and more. And so we basically use this as hard constraints to date our tree. So for example, here's Elismatales on the right, and there are 10 clades that are present in Magellan and Elismatales, our Elismatales. We use those as hard calibrations, date the tree, place it back in. So now we have a dated tree with 590 calibrations for 79,000 species. And then we can do the same thing with the open tree, and we get our dated tree for 356,000. So we can attempt to do things like resolve the polytomy. So a lot of people have talked about this, Yetz et al., Kuhn et al. There are some pros where you can get more reasonable placement of taxa that have no resolution based on birth-death models. There are cons, like you probably shouldn't do diversification analyses because you've already imposed a birth-death model on this. But nevertheless, we can do that. So here's just an example with acrogymnospermies, you might just know it as gymnosperms, and a few iterations of birth-death placement of unresolved taxa. Okay, so how about data overlap? So for each clade in the tree, we can measure how much data overlaps on the left and the right. So basically, in this simple example, you get 8 nucleotides overlap between it, just a raw measure. So if we take Erythales, for example, and look at this, you can see areas that are in red have basically one gene, and areas that are blue have multiple genes. So we can look at this across the whole clade, and it's really nice. It looks really red. The reason why it looks so red is because actually acrogymnospermies has almost whole genomes that overlap. So it's basically like taking the whole color spectrum and making it red. So it's a little bit hard to see, but here's a distribution. And this is number of nucleotides. So 1,700 is the median. That's roughly one gene, maybe 2 genes. So most of the data overlap here is 1 or 2 genes. There are some that are quite large. You can see they're 29,000. What about support? So we can't really do bootstraps. Bootstraps are bad for big trees. Bayesian analysis is just not going to happen. So we can do some other things like A-Big or SH-like, but we chose to do something called quartet concordance. Again, this is something similar to the ICA scores of Salicos and Rocus, using quartets. You can ask me about that later if you want to know more about that. Anyway, but same idea. Here we have low support in red, high support in blue. Here is the whole tree. Again, it's completely unreadable, but if you zoom in, you can see it's again completely unreadable, but it's quite variable. Nevertheless, the score here goes from -1, where some other tree is completely supported, then some other resolution is completely supported than what we get, to 1, our resolution is what the data supports, and the median is about 0.29. So a little bit of support for what our tree says. So you get data. So why are there not more taxa? So I'm just going to rush through this. So we could probably get more taxa. We could probably lose some of the constraints we have, but we have problems with barcoding. So all of the barcoding sequences that have gone on, a lot of them are good, a lot of them are bad. So here's a constraint tree, and you can see some taxa jetting out here. Well, that's with a constraint tree, they jet out. Unconstrained tree, they go completely the wrong area. So they're completely misidentified. So if we were to run an unconstrained tree, we would see this enormous mess. So our constraints actually help us identify misidentified taxa, but it's hard to solve. The other problem is a little bit simpler, which is just that if you use maximum likelihood, barcode sequences could be very similar, and so they have a distance of 0 all over the tree, and that's kind of difficult to deal with, but if you incorporate uncertainty, it's easier. So one problem is that there's little that can be done for this on NCBI side because the original submitter has to fix the data. So that's one issue. The last thing I want to fall back on here is just to show you that you can do other analyses with this now. So here's just a basic analysis of rates of diversification using Medusa. I know all of the controversy around this kind of stuff. I don't want to talk about it, but nevertheless, here is, that was perfect. Here is just an analysis of Medusa on this data set, and I just want to thank my collaborators and co-authors. So, thank you. It is the last talk, so if you want to talk.